Hello everyone, I'm Kevin.
I'm Nia.
I'm Sarah.
And I'm Marco.
And today we'll talk about how we find or use computation to find organelles in cells. When we talk about a cell, we usually talk about its function and most importantly, its organelles. Recent technological advancements have allowed us to find and locate them. They will discuss how computation has helped us accomplish that task.
So before we discuss the computational methods used to locate organelles in cells, we should first review how organelles are located traditionally. So scientists first mark their samples with stains and dyes that bind to specific subcellular structures. Some of the stains used cause color changes that can be viewed under regular white light. Other stains are fluorescent and cause targeted areas to fluoresce under special lighting conditions. Another way subcellular structures are labeled is by using fluorescent proteins that bind to specific targets. Samples marked with fluorescent methods must be viewed under the fluorescent microscope.
There are four main light sources used on fluorescent microscopes: xenon arc lamps, mercury vapor lamps with excitation filters, lasers, and high-power LEDs, which are used to illuminate the samples. This light is passed through a filter before it reaches the sample to make sure that no external light hits it. The stain markers in the samples fluoresce when they absorb energy from the light source, emitting a longer, lower energy wavelength light. This light is separated from that of the brighter light source using a filter, the end result being that only the fluorescent substances are visible. This only lets you take an image of a single color at a time. However, a microscope can contain multiple different filters, allowing for a sample to be stained with multiple dyes that can be viewed separately. This allows for multiple images of different single colors to be taken and consolidated into a more complex image containing multiple colors, as you can see on this image of a human cancer cell. The DNA, microtubules, and a protein called INCENP are stained blue, red, and green respectively. Each image was captured individually, then overlaid to create the final product.
Fluorescent microscopes aren't the only microscopes that we can use to find organelles. There are a lot of possible microscopes to use, and the kind of microscope chosen is very important. For example, using phase contrast microscopes might seem appealing since they allow us to look at live unstained cells and observe the way they move around. Unfortunately, this only provides information about groups of cells moving together to accomplish a certain task in the body. We previously mentioned fluorescent microscopes, which are also powerful, but to look at cells in even more detail, we must use electron microscopes.
Electron microscopes function by taking advantage of the electron's wave properties and reflect back clear images. The downside to this method is that the cells have to be dead. Scanning electron microscope provides an excellent way to analyze the surface of cells, as well as cilia and flagella. This is important because the surface of cells is often filled with lots of important receptors and transport proteins. But to observe the inside of the cell, we will need to use a transmission electron microscope. This microscope involves firing an abundance of electrons on the specimen, which allows each organelle to absorb and reflect different wavelengths using their different absorbance patterns, which enables us to look at the organelles and analyze them.
When using optical or electron microscopy, once the proper structures are labeled, they can be viewed under the microscope and we can visually identify their locations. The stains or markers used highlight the specific structures and allow them to be distinguished from other parts of the cell. In some cases, different organelles can also be distinguished by their shape or size.
An important factor to consider when locating organelles in cells is whether the cell being studied is prokaryotic or eukaryotic. Prokaryotic and eukaryotic cells vary in cellular structure, so the process for identifying organelles in them can differ. Prokaryotes are generally smaller than eukaryotes, which could present some challenges when viewing them, even under microscopes. In addition, scientists often target different parts of the cell when working with prokaryotic and eukaryotic cells. Generally, prokaryotes only have cell membranes, cell walls, and ribosomes, while eukaryotes have a much larger range of organelles, including nuclei.
All said methods are practical and yield excellent results, yet several flaws needs to be addressed. There might be some bacteria that have entered the cell through phagocytosis. Sometimes said bacteria or even other organelles might look like other organelles because they have similar absorbance pattern compared to that one. Using computational methods allow us to avoid this issue.
By analyzing the DNA strands of the proteins created by the organelle, we can easily detect extracellular organisms like bacteria and viruses, especially if we add control samples. Another problem is the lengthy process of analyzing everything by hand. Often, labs spend most of their time analyzing each organelle and verifying for errors. Computation can be used to drastically improve the efficiency of this process.
So how do we use computation to get around having to manually identify every single organelle? One example of how this can be done is the algorithm created by the Allen Cell Institute.
Before this algorithm can be discussed, it is important to know about classifiers. A classifier is an algorithm that sorts input data into labeled categories. Classifiers help this algorithm train and become progressively accurate.
The Allen Cell Institute's algorithm is a label-free method, meaning you don't have to use fluorescent dyes or any other marker in order to tag your samples. Instead, you input a bright field, or regular white light, image of a field of cells, and the algorithm uses a predictive model to add in 3D subcellular structures. It does this using a deep learning algorithm that they have trained using thousands of images of cell fields with certain proteins fluorescently tagged. The algorithm is trained by feeding it both the bright field and fluorescent images of the same field of cells. It then attempts to predict where certain proteins would be on the bright field. The result is compared to the true fluorescent image, and the algorithm's parameters are adjusted to minimize the mean squared error, or the average square difference between the predicted image and the actual one.
Over time, the predictive model learns the relationship between tagged and bright field images. Once it has gotten good enough at this, it can be used to reliably predict where these tagged proteins will be located on a bright field image, even if it hasn't seen that exact image before. After it's added those structures in, the algorithm finishes up by applying cell segmentation, which separates the field of cells into individual ones to allow you to determine the location of subcellular structures in every single cell.
Algorithms that process images aren't the only available option. Another computational method for determining the location of organelles involves locating specific proteins in the cell. First, we need to look at different ways to analyze the protein composition of cells. For example, the mitochondria and the chloroplast contain DNA of their own. This means that when locating these organelles, it could be useful to study the organelle DNA rather than the nuclear DNA. Some organelles, such as the nucleolus, the ribosomes, the cilia, and the flagella, are mostly tertiary proteins, however, so we can easily find information about them directly in the nuclear DNA.
Others, like the lysosomes and the Golgi apparatus, contain mostly proteins with quaternary structure, meaning that we have to search up the tertiary components of the proteins to analyze them. In the case of the smooth and rough endoplasmic reticulums, as well as the Golgi apparatus, we can analyze its membrane, the cisternae. The main protein components of the cisternae are the P47 and the P97 protein. The gene encoding for it is called NSFL1C, and in humans is located on the 20th chromosome. Information such as those we have already found may dictate the ways certain organelles are coded for, and may help in the process of finding other organelles. Even if we haven't found many organelles using computational methods, we have successfully verified the presence of most of them.
Once we locate proteins in the cell, we can use this information to locate organelles as well. Because organelles have specialized functions, they have unique protein makeups, which allows us to identify specific organelles by their proteins. Monitoring the changes in the location of proteins over time can also be useful, as it gives us information about organelle and protein interaction. It can also show the role of specific organelles and proteins in the cell.
Overall, discovering the internal composition of cells is a process that has plenty of room for development. The process is complicated, but allows us to better understand the fundamental particles of life. Thanks for listening.